So we're going to be finishing up the class now with chapter 27. We're going to be looking at the reproductive system. So first, we're going to talk about the male reproductive system. So the organs of the male reproductive system include the testes, the system of ducts, and these systems of ducts include the ductus deferens, the epididymis, the ejaculatory ducts, and the urethra. So these are all the ducts that are going to be involved in the male reproductive system. We're also going to discuss the accessory sex glands. So these are going to include the seminal vesicles, the prostate, and the bulbourethral glands. And then there's also several supporting structures. So these supporting structures include the scrotum and the penis. So the testes, or the male gonads, these produce sperm and they also secrete hormones. The duct system, this is going to transport and store sperm. It's also going to assist in the maturation of that sperm and also convey the sperm to the exterior. Semen contains sperm plus the secretions that are provided by the accessory sex glands. The supporting structures all have various functions, such as the penis delivers sperm into the female reproductive tract, and the scrotum, which is labeled here, this is going to be a supporting structure that supports the testes. So we're going to be looking a little bit more into detail of the structures of the male reproductive system. So first, we're going to be looking at the scrotum. Remember, this is a supporting structure, and the term scrotum literally means bag. So it's kind of easy to remember that. So this is the supporting structure for the testes. Now it consists of loose skin and underlying subcutaneous layer, and it's going to hang from the root or the attached portion of the penis. So it's going to kind of hang from down here. Now externally, the scrotum actually just looks like a single pouch of skin that's separated into lateral portions by a medial ridge that we call raphe. Raphe, the term, means seam. So this is a seam separating the two lateral portions of the scrotum. Internally, on the other hand, though, we have this scrotal septum that divides the scrotum into two separate sacs. Now, each of these separate sacs contains a single testis. Now, it's not specifically labeled here, but we have one testis here and one testis here. This septum, the scrotal septum, is made up of subcutaneous layer and also a muscle tissue, which is called the dartus muscle. Now, this dartus muscle is composed of bundles of smooth muscle fibers. So it's not going to be skeletal muscle fibers. These are going to be smooth muscle. The dartus muscle is also found in the subcutaneous layer of the scrotum, so it's going to be found in the scrotal septum as well as the subcutaneous layer of the full scrotum. Now, associated with each testis in the scrotum is the cremaster muscle. So that's going to be this big muscle here, coming all the way up, down, all the way to here. Now this is a series of small bands of skeletal muscle that essentially descend as an extension of the internal oblique muscle, and this goes through the spermatic cord and it surrounds the testes. So again, this is made of skeletal muscle. Now, the location of the scrotum and the contraction of its muscle fibers really all work together to regulate the temperature of the testes. Now, normal sperm production requires a temperature about 2 to 3 degrees Celsius lower or below normal body temperature. So the cremaster muscle, with that skeletal muscle that it's made up of, really work to move the scrotum to have the testes a certain distance away from the body to maintain that temperature that's 2 to 3 degrees lower than the normal body temperature. So if the testes are getting a little bit too warm for sperm production, the cremaster muscle is going to relax and allow the scrotum to go a little bit further away from the body. And if it's a little bit too cold, then the cremaster is going to contract and bring the scrotum closer to the body to warm up the testes a bit. So we kind of have that regulation of temperature of the testes that's controlled by the muscles surrounding them, mainly the cremaster muscle. Now the testes, also known as testicles, these are those paired oval glands in the scrotum that we just discussed in the last slide. They will develop near the kidneys in the posterior portion of the abdomen in the developing male, and they usually begin their descent into the scrotum during the latter half of the 7th month of fetal development. So kind of the end of the 7th month of development. This is when they're going to drop from where they develop near the kidneys down into the scrotum. Now here on this picture, I kind of just have the internal view of the scrotum brought up so we can kind of look at the membrane surrounding the testes as well as some of the other structures surrounding them. So there is a serous membrane called the tunica vaginalis. This is derived from peritoneum and it forms during the descent of the testes. And this membrane partially covers the testes. Now, just internal to the tunica vaginalis, the testes are surrounded by this white fibrous capsule that's composed of dense irregular connective tissue, and we call this layer the tunica albuginea. Now, this layer extends inwards and it forms the septa that divides the testes into a series of internal compartments called lobules. And we'll see those compartments in the next slide. But the tunica albuginea is going to be the connective tissue that kind of invaginates the testes and divides it further into these compartments called lobules. Now, each testis contains about 200 to 300 lobules, and these each contain 1 to about 3 of these tightly coiled tubules called seminiferous tubules. And we'll see this on the next slide. This is where sperm is produced. So here on this slide, we have this sagittal section of the testes. We can see the tunica vaginalis as that external connective tissue layer, and then the tunica albuginea as that internal connective tissue layer that separates the testes into these smaller compartments called lobules. So again, each testis has approximately 200 to 300 of these lobules. So we'll put this right here, about 200 to 300 in each testis. And each of these lobules contains 1 to 3 of these really tightly coiled tubules called the seminiferous tubules. So that's going to be each of these. You can see these really coiled seminiferous tubules here. And this is going to be the actual location of sperm production. Now, the process by which these seminiferous tubules actually produce sperm, this is called spermatogenesis. We will definitely go over the entire process of spermatogenesis yet, but this is just the process of sperm production. Within these seminiferous tubules, we have two major types of cells. These are going to be spermatogenic cells and sustentacular cells. Now, these spermatogenic cells, these are going to be the actual sperm-forming cells. And the sustentacular cells have a lot of functions, but they really just function to support spermatogenesis. Most of their functions all work to support the production of sperm. Sustentacular cells are also called Sertoli cells, so if you see these in your textbook or any of your additional readings, these are the same cells, but we're going to see them a little bit closer on the next slide. But both of these cells, spermatogenic cells and the sustentacular cells, are found here in those seminiferous tubules. Okay, on the right here, we can just kind of see a real life kind of cadaver view, an internal view of the testes. We can see the testes in there with the tunica vaginalis, and then a more internal view, sagittal section on the right side, showing us the tunica albuginea and those seminiferous tubules. They're always more difficult to see on these cadaver sections, but definitely cool to kind of see not just a cartoony picture, but an actual real life example here. Okay, now on the left here, we have a really close-up view of a transverse plane through the seminiferous tubules. So kind of here, above here, you can kind of see this section. We're getting this big cartoony picture just from here, right inside those seminiferous tubules. So we can kind of see these individual cells, what they look like, and how they interact with one another. So just to kind of orient you with these cells, on the left we have this labeled the Sertoli cells. Remember, Sertoli cells is another name for those sustentacular cells, which are those supporting cells. And then on the right here we have all these labeled spermatogenic cells. So these are all going to be different forms of spermatogenic cells through the development of sperm. So from bottom to top, we're going to go through each of these different forms of the spermatogenic cells. So first at the bottom here, we have stem cells that are called spermatogonia, or plural would be spermatogonium. And these develop from primordial germ cells that arise from the yolk sac, and these are actually going to enter the testes during the 5th week of development. That's when these stem cells kind of enter into the testes. So now we kind of have this order of advancing maturity. So we're going to be starting from the stem cells here at the bottom, the spermatogonium, and as these sperm cells mature and they go through these different forms, different cycles of their life, and they get more and more mature, we have different names for them. So the first stage of maturity, they're going to be called primary spermatocytes, kind of cut off on the right there, but these are primary spermatocytes. Then we have secondary spermatocytes. They turn into spermatotids, early and late spermatotids, and then eventually they become the most mature that they can, and then they're called sperm cells. After a sperm cell has formed, it's going to be released into the lumen of the seminiferous tubules. So kind of this area here is going to be the lumen inside the seminiferous tubules. So we kind of go from least mature down here at the basement membrane, and as they kind of move up closer and closer to the lumen of the seminiferous tubules, they mature. They gain a lot of function, they change their shape, they change their function, they mature into these functional sperm cells. So you can see these cells here, these spermatogonia, into primary spermatocytes, secondary spermatocytes, and then kind of here we're kind of the spermatids, and then this guy here is going to be a sperm cell. Now you'll also notice these Sertoli cells or these sustentacular cells. these are embedded among these spermatogenic cells. Now these are going to be these large cells here. They kind of fully surround all of these developing spermatogenic cells. So we can see them here. This is a pretty big cell, kind of surrounds all of these cells. So it's going to extend from the basement membrane all the way to the lumen of the tubule. Now, internal to the basement membrane and the spermatogonia, we're going to have tight junctions that join these neighboring sustentacular cells to one another. So you can see these kind of right here. Maybe I'll choose a better color here, maybe this blue, but there's a tight junction here and here and here. So these are tight junctions that are connecting those sustentacular cells to one another. These junctions form this obstruction that is known as the blood-testis barrier. Now, it's called this because substances must first have to pass through the sustentacular cells before they can reach the developing sperm. So they kind of create this barrier to protect the sperm from anything coming through the blood capillary right here. Now, these sustentacular cells will support and protect the developing spermatogenic cells in several ways. They're going to nourish these spermatocytes. So nourish them. It'll really nourish these spermatogenic cells at every stage in their development. It's also going to phagocytize excess spermatid cytoplasm as the development proceeds. So we're going to phagocytize, kind of garbage essentially, kind of this extra stuff that's produced during development. Phagocytize. It's also going to control the movements of these spermatogenic cells. It's kind of like make sure that they go in the right area, that they're always going to be moving towards the lumen of the seminiferous tubules as they develop. So control their movement. And they're also going to really help to release the sperm into the lumen of the seminiferous tubule. So this is kind of along with controlling that movement. They also produce fluid for sperm transport. Produce fluid. So this is just going to be essentially fluid for helping them moving within the seminiferous tubules. The fluid that the sustentacular cells produce to help the sperm move around is not really incorporated into semen at all. It's just to help them move through their development from the spermatogonia into sperm cells. These sustentacular cells will also secrete the hormone inhibin. And they also will regulate the effects of testosterone and follicle stimulating hormone. So regulate testosterone and the follicle stimulating hormone. So here we can see another good picture of the sustentacular cells and how they kind of are completely surrounding these spermatogenic cells through their development. So this is essentially the whole process of spermatogenesis. So in humans, spermatogenesis is actually going to be a process that takes about 65 to 75 days. This process is going to begin with the spermatogonia, which contains diploid number of chromosomes, so they are going to be 2n. And remember, these spermatogonia are a type of stem cell. Now, when these spermatogonia undergo mitosis, some spermatogonia will remain near the basement membrane, and they're going to be in this undifferentiated state, and essentially they serve as a reservoir of cells for future cell division and subsequent sperm production. So essentially, if we have one stem cell and it's going to divide, it's going to go through mitosis, divide into two cells. One of those cells is going to be a primary spermatocyte. I'll show this in blue. And the second cell that it divides into is going to be another spermatogonia, so that this cell can divide again if it wants to later. It can go through mitosis. This cell can't go through mitosis again. So once it differentiates into a primary spermatocyte, it can't go through mitosis again. It's only these stem cells, these spermatogonia, that are able to go through mitosis. So whenever these spermatogonia go through mitosis, they're always going to divide into a differentiated cell, in this case a primary spermatocyte, and an undifferentiated cell, so that we kind of continue to have this reservoir of cells that can continually divide down the road. The rest of the spermatogonia are going to lose contact with the basement membrane once they differentiate into these primary spermatocytes. Then they lose that contact with the basement membrane, and they squeeze through the tight junctions of the blood-testis barrier. They're going to undergo these developmental changes, and once they kind of leave the basement membrane, they go through these developmental changes. It's at this point that they are considered primary spermatocytes and no longer spermatogonium. They have to differentiate. They have to go through these developmental changes to kind of show that they are for sure on this path of developing into sperm. They're no longer a stem cell. They have decided that I'm going to be a sperm cell and I'm going to go through that developmental path. Primary spermatocytes are also diploid, so they are 2N. So remember, this diploid 2N in this case means that they have 46 chromosomes. Now, shortly after it forms and it differentiates, each primary spermatocyte is going to replicate its DNA, and then that's when meiosis begins. So meiosis 1, this is where we have the homologous pairs of chromosomes line up at the metaphase plate, and then crossing over occurs. So I think we're going to see this in the next slide in picture form, but I'll write it down right here for you guys too. So meiosis 1, this is when we have lining up at the metaphase plate. This is the chromosomes. Remember, this is homologous pairs of chromosomes. So remember, the homologous pairs of chromosomes, this means that they have the same structural features and the same pattern of genes. They don't necessarily have the exact same genes, but they have the same pattern of genes. So they will line up at the metaphase plate, and then this is also where crossing over occurs. So then the mitotic spindle pulls one chromosome of each pair to an opposite pole of the dividing cell. The two cells that are formed by meiosis 1, these are called secondary spermatocytes. So this is the result of meiosis 1. Now, each of these secondary spermatocytes has 23 chromosomes, so they are haploid. So they are 23 chromosomes. Each chromosome within a secondary spermatocyte, though, is made up of 2 chromatids, so they are still attached by a centromere. So at this point, after meiosis 1, we still have 23 chromosomes, each with 2 chromatids. So remember, this is 1 chromatid and a second chromatid. Now there's going to be no replication of DNA that occurs in the secondary spermatocytes. In meiosis 2, which is kind of going to be the next step here, this is when the chromosomes are going to line up in a single file, again, along that metaphase plate. And the 2 chromatids of each chromosome are going to separate. So this chromatid is going to go one way and this chromatid is going to go the other way. Now there are going to be 4 haploid cells resulting from meiosis 2, and these are now termed spermatids. So as we kind of go through this, we kind of realize that a single primary spermatocyte is going to produce 4 spermatids, and this is as a result of 2 rounds of cell division, meiosis 1 and meiosis 2. So here in this figure, we can see these events in spermatogenesis. Now remember that the diploid cells, 2 end cells, these are going to have 46 chromosomes. And haploid cells, which are just N, these have 23 chromosomes. So here at the top, again, we start with these spermatogonium. I know we're just going through this entire process again, but it has to be kind of solid in your brain. So at the top, we have these spermatogonia. They are stem cells which can differentiate or choose their path in life into a primary spermatocyte. So here we have this differentiation. Essentially, this is just choosing the role and saying, this is what I'm going to do, this is what I'm going to develop into. If they're undifferentiated, that means they are essentially haven't picked their role in life yet. They haven't chosen which path they're going to take, and so they can kind of take multiple paths if they are undifferentiated. Once they are differentiated, they have chosen their path, and that is the developmental path that they must take. So once they differentiate, they are a primary spermatocyte. Now this primary spermatocyte is going to go through meiosis 1 to produce secondary spermatocytes. Now one really interesting and pretty unique process happens during spermatogenesis. This is pretty unique to spermatogenesis. As the spermatogenic cells will proliferate or divide, they really fail to complete the cytoplasmic separation. So they don't fully complete cytokinesis. What this means is that essentially the cells remain in contact with each other by these cytoplasmic bridges. So we can see these little bridges of cytoplasm between these divided cells. And so they keep these cytoplasmic bridges throughout their entire development. So we can see them here and we can see them here. We have it labeled over here, those cytoplasmic bridges. And they really only lose those cytoplasmic bridges when they are completely mature, completely developed into kind of the end of their development path. Now, the final stage of spermatogenesis after meiosis 2 produces 4 spermatids. Now we've reached kind of this final stage of spermatogenesis, which is spermiogenesis down here. So this is the development of these haploid spermatids into the actual final developed sperm cells. So these are called sperm cells or spermatozoa. Now, in spermiogenesis, there's not really any division that occurs. It's just kind of this final developmental stage where they lose those cytoplasmic bridges and where each of those spermatids becomes a single sperm cell. So during this process, these very spherical spermatids will transform and develop into these very elongated and slender sperm cells. Each day, about 300 million sperm in a male complete the process of spermatogenesis. They contain several structures that are really highly adapted for reaching and also penetrating a secondary oocyte in the female reproductive system. The major parts of a sperm are the head and the tail. Now, the head contains a nucleus with 23 really highly condensed chromosomes. Covering the anterior 2/3 of the head is the acrosome. Now, the acrosome is kind of a cap-like vesicle that's filled with enzymes that help the sperm to penetrate a secondary oocyte and to bring about fertilization. The neck is considered the region just behind the head, and this is going to contain the centrioles. The middle piece contains mitochondria. And these mitochondria are arranged in a spiral, and they provide all the energy or the ATP provided for locomotion. The principal piece is the term that we use for the longest portion of the tail. And the end piece is the terminal or the tapering portion of the tail. And we're going to see all these in a picture on the next slide. So here in this picture, we can see a typical sperm cell. We have the head and the tail, which are the two major components. We have the acrosome here at the top. This is this apical vesicle, and remember, it's going to be filled with enzymes. And these enzymes are going to help the sperm to fertilize an egg. Here you can also see a really huge concentration of mitochondria within this middle piece. Remember, these mitochondria produce a ton of ATP for locomotion. Once ejaculated, most sperm don't survive more than really like 48 hours. And most of this time, again, this is 48 hours within the female reproductive tract. And because of this, they have to act rather quickly. They have to move very quickly. Those mitochondria have to produce a ton of ATP in order for them to move very quickly through the female reproductive tract in order to fertilize the secondary oocyte. So this 48 hours is within the female reproductive tract. The female reproductive tract is not very hospitable towards sperm, and so they will die off rather quickly, like I said, within 48 hours of being within the female reproductive tract. Now we're going to kind of move on and talk more about the hormones that are at play. Now, the initiating factors are kind of unknown for puberty, but at puberty, there are certain hypothalamic neurosecretory cells that increase their secretion of gonadotropin-releasing hormone. So this is GnRH. And this hormone is going to stimulate gonadotrophs in the anterior pituitary gland, and it's going to stimulate them to increase their secretion of two gonadotropins. Now, these two gonadotropins are the luteinizing hormone, remember we also call this LH, and the follicle stimulating hormone. Now, LH, the luteinizing hormone, this is a hormone that stimulates cells in the testes to produce testosterone. The follicle stimulating hormone, on the other hand, is going to stimulate spermatogenesis. So this figure here shows the hormones and the negative feedback loops that control the secretion of testosterone and also control spermatogenesis. So let's first look at these green arrows that indicate stimulating secretion. So up here we have the hypothalamus, where those neurosecretory cells are going to secrete the gonadotropin releasing hormone, that GnRH. Now remember that gonadotropin releasing hormone is going to stimulate gonadotrophs in the anterior pituitary to secrete both the follicle stimulating hormone and luteinizing hormone. Now these two hormones will do different things, and we will talk about both of them. First, we'll go to the right side here to this luteinizing hormone. So luteinizing hormone is going to stimulate testosterone secretion. And it does this by stimulating these cells, which are called interstitial cells. Interstitial cells, I don't think we've really talked about these cells yet, but they are cells that are located between the seminiferous tubules within the testes. And these interstitial cells secrete the hormone testosterone. So we see testosterone going this way and also this way in this diagram. Testosterone, this is a steroid hormone, and it is synthesized from cholesterol in the testes, and it is a principal androgen. So this is just the type of hormone that it is. Testosterone is lipid soluble, and it very readily diffuses out of the interstitial cells into the interstitial fluid and then into the blood. So now we have this testosterone from the interstitial cells. Now, in response to stimulation from follicle stimulating hormone and also the testosterone, we have sustentacular cells that are stimulated to secrete androgen binding protein. So this is called ABP. ABP is really important. Its main function is to initiate spermatogenesis. Now, at the bottom of this diagram here, we have another hormone. This is dihydrotestosterone, also called DHT. Now, in some target cells, like those in the external genitals and the prostate, there's this enzyme called 5-alpha reductase. You won't really have to remember that enzyme, but it's going to actually convert testosterone into another androgen, which is called dihydrotestosterone. Testosterone and dihydrotestosterone will both bind to the same androgen receptors, and these are found within the nuclei of target cells. Remember, because these are lipid soluble. Now, the hormone receptor complex regulates gene expression. Remember, these will turn some genes on and turn others off. Now, because of these changes, the androgens will produce several effects. The effects that these androgens produce, we will actually talk about on the next slide, because while we're on this slide as well, I do kind of want to talk about these negative feedback loops. Quickly though, that dihydrogen testosterone will produce these male pattern of development, enlargement of the male sex organs, and these male secondary sex characteristics starting at puberty, and then also anabolism. But we'll go into that a little bit later. Okay, so we also have these negative feedback loops, these red dashed lines. So this is indicative of inhibition. Both ABP and testosterone will work within these negative feedback loops to inhibit gonadotropin releasing hormone secretion when there are significant amounts of ABP or testosterone. So you can see this red dotted arrow here on the right side. This is the testosterone decreasing the release of the gonadotropin releasing hormone at the top there, so it's inhibiting further release when there are large amounts of testosterone present. And when there's large amounts of testosterone, we're also going to have inhibition of luteinizing hormone. So kind of those two separate negative feedback loops. On the other side here, we have those sustentacular cells and how they're involved in negative feedback loops and inhibiting further secretion of follicle stimulating hormone. So when there are large amounts of ABP present, these sustentacular cells are going to secrete inhibin. And inhibin is actually going to decrease the release of follicle stimulating hormone. So we kind of get this negative feedback loop from those sustentacular cells, and all these negative feedback loops are going to decrease the secretion of GnRH, FSH, and also LH. Okay, so in this slide, we're going to go over what those effects of testosterone and dihydrotestosterone are going to have on the male body. So these are both androgens, and essentially they have very similar effects, so we're just going to kind of lump them together and talk about them in the same kind of category. So these androgens will produce several effects here. And again, those effects are produced by binding to a receptor within the nuclei, and then regulating gene expression by either turning some genes on or others off. So the first kind of effect that they can have is prenatal development. So this is before birth. Testosterone is going to stimulate the male pattern of development of reproductive system ducts and also the descent of the testes. Remember, we talked about that coming in the 7th month of development. We're also going to see the development of male sex characteristics. So this occurs at puberty, and this is where testosterone and dihydrotestosterone are going to bring about the development and enlargement of the male sex organs, and also that development of the masculine secondary sex characteristics. So I haven't really talked too much about secondary sex characteristics. But these are going to be traits that distinguish males and females, but they don't have a direct role in reproduction. So in males, secondary sex characteristics are going to be things like a deeper voice or things like a beard. These are going to be examples of secondary sex characteristics in males. These androgens will also stimulate or also have an effect on the development of sexual function. So androgens will contribute to male sexual behavior as well as spermatogenesis. And androgens also have a role in sex drive in both males and females. These androgens will also stimulate protein anabolism. So androgens are anabolic hormones. So what this means is that they stimulate protein synthesis. We know that negative feedback systems regulate testosterone production. So here we have a negative feedback diagram. So at the top here, when testosterone concentration in the blood increases to a certain level, then this is when it's going to inhibit the release of GnRH by cells in the hypothalamus. So this is here, these cells in the hypothalamus will decrease their secretion of gonadotropin releasing hormone. Now, as a result, there's going to be less GnRH in the portal blood that flows from the hypothalamus to the anterior pituitary. Now, these gonadotrophs in the anterior pituitary will then release less LH, less luteinizing hormone, so that the concentration of luteinizing hormone in the systemic blood is going to decrease. So that's going to be the output here. Now, with less stimulation by luteinizing hormone, the interstitial cells, or the Leydig cells, in the testes are going to secrete less testosterone. And because of this, we're going to get a decrease in the overall testosterone level in the blood, and we're going to get a return back to homeostasis. On the other hand, if the testosterone concentration in the blood falls too low, like if the response down here is too strong and we get too much of a decrease in blood testosterone, this is when the GnRH is again going to be released by the hypothalamus. It's going to stimulate the secretion of luteinizing hormone by the anterior pituitary, and then that consequently is going to stimulate testosterone production in the testes. So let's talk about the duct system in the male reproductive system. So we have these sustentacular cells producing fluid, and this is going to create a pressure-driven force. So this is going to be pressure generated by the fluid secreted by the sustentacular cells, and this pressure is going to push the sperm and fluid along the lumen of the seminiferous tubules, and then into a series of very short ducts, which are called the straight tubules. The straight tubules will then lead into a network of ducts in the testes called the rete testes. And from the rete testes, the sperm will move into a series of very coiled efferent ducts. And this is going to be in the epididymis. And these efferent ducts are going to all empty into a single tube called the ductus epididymis. We will see these all on the next diagram, but here we'll just have it all in written form. So the efferent ducts are going to go empty into a single tube, which is called the ductus epididymis. Then the ductus epididymis turns into the ductus deferens as it kind of moves up into the spermatic cord. Now the epididymis, this is another term that we need to know, just epididymis. This is a comma-shaped organ, and it lies along the posterior border of each testis. Now, each epididymis really consists mostly of those tightly coiled ductus epididymis, but it's kind of just made up of some of the duct system. So here on this diagram, we can see we have labeled the head of the epididymis, the body of the epididymis, and then the tail of the epididymis. So kind of like I said, it's kind of like a comma-shaped organ. So it kind of moves like this, down to here, and then kind of essentially there is where we're going to have the epididymis organ. Now functionally, the epididymis is going to be the site of sperm maturation. So this is the process by which the sperm actually acquire motility, or the ability to move, and they also acquire the ability to fertilize an ovum in this area. Now this maturation period really occurs over a period of about 14 days. The efferent ducts from the testis will join the ductus epididymis. At the larger superior portion of the epididymis, the head, Then we have the body. This is going to be a more narrow mid portion of the epididymis, and then the tail is that smaller inferior portion. At the distal end, the tail of the epididymis is going to continue up as the ductus deferens. We also call this the vas deferens. Now we know that the ductus epididymis is going to be very coiled, not very straight at all, but within the tail of the epididymis, the ductus epididymis becomes less convoluted and the diameter also increases. And then beyond this point, where we get a little bit straighter in the tube and the diameter becomes larger, this is where the duct is known as the ductus deferens or vas deferens. Now the ductus deferens is going to ascend along the posterior border of the epididymis through the spermatic cord and then will enter into the pelvic cavity. There it's going to loop over the ureter and pass over the side and down the posterior surface of the urinary bladder. So we see that all here in this figure. So here we can see the ductus deferens as it comes up and over the urinary bladder. In this diagram, we can really see the locations of several accessory reproductive organs in males as well. So here we can see the prostate, which is going to be this organ here, or a gland rather. We can see the urethra. We will talk about the different parts of the urethra as well in males, but this is going to be this tube system here. And we can also see the penis as well, the entire organ here. And these are going to be sectioned to show internal details. We also see this portion here. This is going to be the dilated terminal portion of the ductus deferens, and we call that the ampulla of the ductus deferens. Okay, the spermatic cord. This is a supporting structure of the male reproductive system that ascends out of the scrotum. Now it consists of the ductus deferens as it ascends through the scrotum. It also contains the testicular artery, the veins that drain the testes and also carry testosterone into circulation. The spermatic cord also contains the autonomic nerves, lymphatic vessels, and also that cremaster muscle. Now the spermatic cord and the ilioingual nerve will pass through the inguinal canal. And this is a kind of a small passageway in the anterior abdominal wall, and it kind of carries that spermatic cord, or allows the spermatic cord and the nerve to pass from the scrotum into the abdomen. Each ejaculatory duct is formed by the union of the duct from the seminal vesicle and the ampulla of the ductus deferens. The short ejaculatory ducts form just superior to the base of the prostate, and they pass inferiorly and anteriorly through the prostate. In males, the urethra is the shared terminal duct of both the reproductive and the urinary systems. So what this does is that it serves as a passageway for both semen and urine. The urethra passes through the prostate gland, the deep muscles of the perineum, and also the penis. Now this figure shows the internal structure of the penis. We have the prostatic urethra up here. This passes through the prostate. Now, as this duct continues inferiorly, it passes through the deep muscles of the perineum, and this is where it's known as the intermediate urethra. This is also known as the membranous urethra. Now, as the duct passes through the corpus spongiosum, down here, this is known as the spongy urethra. Now, the spongy urethra, this is going to be the longest portion of the male urethra, and it ends at the external urethral orifice. So here we have those three segments on the right: prostatic, intermediate or membranous urethra, and the spongy urethra. So we've mostly covered the ducts of the male reproductive system. Remember, these are going to be the ducts that store and transport sperm cells. And now we're going to kind of move on to the glands. So the accessory sex glands, these are going to secrete most of the liquid portion of semen. The accessory sex glands include the seminal vesicles, the prostate, and the bulbourethral glands. So we'll talk about the first two on this slide, and then the bulbourethral glands we'll look at on the next slide. So first off, we have the paired seminal vesicles, also known as seminal glands. These are quite convoluted pouch-like structures, and they lie posterior to the base of the urinary bladder and just anterior to the rectum. Now, through the seminal vesicle ducts, they secrete an alkaline, quite viscous fluid, and this fluid contains fructose, which remember is a monosaccharide sugar. It also contains prostaglandins and clotting proteins that are different than the clotting proteins that we find in the blood. Then we have the prostate. This is a single donut-shaped gland. It's about the size of a golf ball, and it secretes a milky, slightly acidic fluid that contains several substances. So slightly acidic, so we're talking about a pH of about 6.5. So prostatic fluid contains citric acid, proteolytic enzymes, acid phosphatase, and then seminal plasmin. So the citric acid in prostatic fluid is used by sperm for ATP production, and they use the Krebs cycle to produce that ATP. These proteolytic enzymes like PSA, pepsinogen, lysozyme, amylase, these are all going to work to eventually break down the clotting proteins from the seminal vesicles. The function of acid phosphatase secreted by the phosphate is kind of unknown. We won't really need to go into that. But then seminal plasmin, this is an antibiotic fluid that can actually destroy bacteria. Then we have the bulbourethral glands, also known as the Cowper's glands. These are located just inferior to the prostate on either side of the membranous urethra within those deep muscles of the perineum. The glands of the bulbourethral glands are going to open into the spongy urethra. Now, during sexual arousal, the bulbourethral glands will secrete an alkaline fluid into the urethra that protects the passing sperm by neutralizing acids from urine in the urethra. These glands will also secrete a mucus that lubricates the end of the penis and the lining of the urethra. And this also decreases the number of sperm that are damaged during ejaculation. Semen, this is a mixture of sperm and seminal fluid. Remember that seminal fluid is a liquid that consists of the secretions of the seminiferous tubules, the seminal vesicles, prostate, and the bulbourethral glands. So this fluid consists of the liquid secreted by all these glands. Now, despite the slight acidity of the prostatic fluid, semen has a slightly alkaline pH of approximately 7.2 to 7.7, And this is due to the higher pH and the larger volume of fluid from the seminal vesicles. The seminal fluid really has a really important function. It provides sperm with a transportation medium, it provides nutrients to the sperm, and it also provides protection from the really hostile acidic environment of the male's urethra and also the female's vagina. The penis contains the urethra and is a passageway for the ejaculation of semen and the excretion of urine. In the next diagram, or in the next slide, we will see a diagram, but the body of the penis is composed of 3 cylindrical masses of tissue, which are each surrounded by a fibrous tissue called the tunica albuginea. The 2 dorsolateral masses are called the corpora cavernosa penis, and the smaller mid-ventral mass is called the corpus spongiosum penis, and this contains the spongy urethra that keeps the urethra open during ejaculation. The distal end of the corpus spongiosum penis is a slightly enlarged acorn-shaped region called the glans penis. And covering the glans in an uncircumcised penis is the loosely fitting prepuce, also known as the foreskin. So here we have that cross-sectional diagram. So skin and a subcutaneous layer enclose all 3 masses of the penis, which consist of erectile tissue. Erectile tissue is composed of numerous blood sinuses, remember, which are vascular spaces, which are lined by endothelial cells and surrounded by smooth muscle and elastic connective tissue. In these figures, you can also see the tunica albuginea of each cavernosa of the penis. So we have a corpora cavernosa on each side. Remember, these are these larger masses on either side here, so one on the left and one on the right. And you can also see the tunica albuginea of the corpus spongiosum. And the corpus spongiosum is that single mass on the ventral side here, surrounding the urethra. So remember that tunica albuginea is the fibrous tissue that surrounds the 3 masses of tissue in the penis, the corpora cavernosa penis and the corpus spongiosum penis. Okay, we're going to be moving on to the female reproductive system now. So the organs of the female reproductive system include the ovaries, which are the female gonads, the uterine tubes, also known as the fallopian tubes or oviducts, the uterus, the vagina, and the external organs, which collectively are called the vulva. The mammary glands are considered part of both the integumentary system and also the female reproductive system. So this figure here shows us a sagittal section showing the female organs of the reproductive system and the surrounding structures. So we can see the distal ovary in this figure here. You can also see the uterine tube off of that same ovary coming right here. But you can also see the positioning of the bladder, which is going to be here, labeled here. We can see it in the diagram here. This is going to be the urinary bladder, and we can see its position between the uterus, just above it here, and also the positioning of the rectum in relation to both those. So here we have the rectum and anus here. The ovaries, which are the female gonads, are paired glands that essentially kind of resemble unshelled almonds in size and shape, and these are going to be homologous to the testes. Remember that homologous here means that the two organs have the same embryonic origin. Ovaries produce gametes. The gametes are going to be these secondary oocytes that will develop into mature ova, also known as eggs, during fertilization. And ovaries also produce hormones. So these hormones will include progesterone and estrogens, which estrogens are going to be the female sex hormones. And the ovaries will also produce inhibin and relaxin. We will go into these hormones in a bit more detail later, and we will also go into the maturation of the female gametes a little bit later as well. The ovaries are supported by a few different ligaments. So we have the broad ligament, the ovarian ligament, and the suspensory ligament, So here we have a superior view of a transverse section. You can see the ovaries. We have paired ovaries, one on either side of the uterus. We have the uterus here in the middle. And these ovaries will descend to the brim of the superior portion of the pelvic cavity during the 3rd month of development. Now, like I said, there's going to be a series of ligaments that holds these ovaries in position. So the first one, like I said, is going to be the broad ligament. So use a different color here, but here we kind of have this broad ligament kind of right here on either side, attaching the ovaries to the uterus. Then we have the ovarian ligament here. This is a very large ligament, and this is going to anchor the ovaries also to the uterus. Just give it a second here with this color. So here is going to be these large anchor points there. And then lastly is the suspensory ligament. So this ligament is back here. And this ligament is actually going to be attaching the ovaries to the pelvic wall. Okay, so we will see this in a diagram in a few slides, but histologically, the ovaries consist of the following parts, or each ovary does. First one we have is the germinal epithelium. This is a layer of simple epithelium. Usually it's low cuboidal or squamous epithelium, and this is going to cover the surface of the ovary. As we kind of go through these layers, we're kind of going to go from superficial to deep layers as we kind of go down. Okay, the next layer is the tunica albuginea. This is going to be a whitish capsule of dense irregular connective tissue. Again, it's located just immediately deep to the germinal epithelium. Then we have the ovarian cortex, just deep to the tunica albuginea. This layer consists of ovarian follicles, which are surrounded by dense irregular connective tissue. And this dense irregular connective tissue contains collagen fibers and these fibroblast-like cells, which are called stromal cells. Again, we'll go into this in a little bit more detail a little bit later. Lastly, we have the ovarian medulla. This is just deep to the cortex. And the border between the cortex and the medulla is not really distinct, but the medulla consists more of loosely arranged connective tissue. And in the medulla, we do find blood vessels, lymphatic vessels, and nerves that we don't really see in the cortex. Okay, so I'm going to kind of go into a little bit more detail about these ovarian follicles. We'll see pictures of them yet, but these are in the cortex and they consist of oocytes in various stages of development as well as the cells that surround them. Now, when the surrounding cells surrounding the oocyte form a single layer, they're called follicular cells. So single layer of cells surrounding the developing oocyte. These are going to be called follicular cells. And then later in development, when they form several layers, these are referred to as granulosa cells. So again, this is going to be later in development, but they form many, many layers surrounding these developing oocytes. But the oocytes, as well as the cells that surround them, collectively are called ovarian follicles. A mature follicle, also known as a Graafian follicle, this is a large fluid-filled follicle that essentially is ready to rupture and ready to expel its secondary oocyte. So this expelling of the secondary oocyte, this is known as ovulation. This is when it releases that secondary oocyte, that egg essentially. Some other terms that we want to kind of go over here, the corpus luteum. This is a structure that contains the remnants of the mature follicle after ovulation. So after it's released that secondary oocyte, a Graafian follicle kind of turns into a corpus luteum. The corpus luteum produces progesterone, estrogens, inhibin, and relaxin. And it'll produce these hormones until it degenerates into a fibrous scar tissue. And once it turns into that scar tissue, we call it the corpus albicans. Here we can see the histology of an ovary. So we can see the germinal epithelium, that most superficial layer. We can also see the ovarian cortex, the ovarian medulla here. And we can also see these various stages of development of the oocyte. So these arrows here, these are going to indicate the sequence of developmental stages that occur as part of the maturation of an ovum during the ovarian cycle. So it's going to start as a primordial follicle, then to a primary follicle, and then as development continues, it will turn into a secondary follicle, then into a mature follicle, remember with that fluid in there, and then into a corpus hemorrhagicum. Now this is when the secondary oocyte right here is going to be discharged. So remember, this is called ovulation. Now once the egg is released, the follicle turns into the corpus luteum and then into the corpus albicans. So this is just kind of a very quick overview of the development of the follicle and the development of the oocyte. The formation of gametes in the ovaries is termed oogenesis. In contrast to spermatogenesis, which begins in the males at puberty, oogenesis actually begins in females before they're even born. Now, oogenesis occurs pretty much in the same manner as spermatogenesis. This is where we have meiosis take place, and then the resulting germ cells will undergo maturation. During early fetal development, like I said, before the female's even born, these primordial germ cells or primitive germ cells will migrate from the yolk sac to the ovaries. Now there, the germ cells will differentiate within those ovaries into oogonia. Now remember, when stem cells differentiate, they're essentially committing to a certain cell type. So at this point, when they differentiate, these cells are committing to becoming oocytes, or committed to becoming egg cells. Oogonia are diploid stem cells, so these are 2N stem cells, and oogonia will divide mitotically to produce millions of germ cells. So these are still stem cells. Now oogonia will divide into germ cells, and some of those germ cells will become primary oocytes. Each of these primary oocytes is going to be surrounded by follicular cells, and this forms a primordial follicle. Now, as we go forward with development within the ovaries, we're going to kind of follow two separate developmental paths. So we're going to follow the development of the oocytes and also development of the follicles. So here we have a primordial follicle. This is where each primary oocyte is going to be surrounded by a single layer of flat follicular cells. Now this entire structure with the oocyte and the follicular cells is called the primordial follicle. Now the ovarian cortex surrounding the primordial follicle consists of collagen fibers and these fibroblast-like stromal cells. You can see these here. So the collagen fibers and the stromal cells are not technically part of the follicle. The basement membrane kind of creates that barrier between the follicle and the ovarian cortex. Now, at birth, approximately 200,000 to 2 million primary oocytes are going to be within each ovary. Now, of these, there's only going to be about 40,000 that are still present at puberty. Now, of those that are present at puberty, there's only going to be about 400 that will mature and ovulate during a woman's reproductive lifetime. So about 400 will mature. So you can tell there's a whole ton of these primary oocytes that don't even make it to maturity and don't ovulate. Now each month after puberty and up until menopause, the gonadotropins FSH and LH, remember, which are the follicle stimulating hormone and luteinizing hormone secreted by the anterior pituitary, are going to continue to be secreted to further stimulate the development of several of these primordial follicles. Though usually there's only about one that will typically reach maturity needed for ovulation. So only one of those will completely reach maturity and be ovulated at the end of every month. So the next step in follicular development is going to be the primary follicle. So here we have kind of a late stage primary follicle. So as a few of those primordial follicles start to grow that we saw in the previous slides, they're going to develop into these primary follicles. Now each primary follicle consists of a primary oocyte that's surrounded by a later stage of development by several layers of these cuboidal and low columnar cells that are called granulosa cells. So you can kind of see there's a lot more cells between that primary oocyte and the basement membrane there. So those outermost granulosa cells will rest on the basement membrane. Now we will talk a little bit about a couple of these other terms kind of in the next slide, but you can still see these collagen fibers and these other stromal cells surrounding the follicle on the other side of that basement membrane. We also have this zona pellucida, which we will go over in the next slide. So as that primary follicle continues to grow, it forms this clear glycoprotein layer that's called the zona pellucida. So that's that clear layer surrounding the primary oocyte that we just saw on that last slide. And this glycoprotein layer forms between the primary oocyte and the granulosa cells. Now, in addition, this new stage of development, we have those stromal cells surrounding the basement membrane. They begin to form pretty much like an organized layer that we now call the theca folliculi. So this is just an organized or more organized layer of those stromal cells that we saw in the primordial follicle. So that's going to be the primary follicle. Now with continuing maturation, that primary follicle is going to develop into a secondary follicle. Now in a secondary follicle, that theca folliculi is going to differentiate into two separate layers, the theca externa and the theca interna. Now the theca externa is going to be that outermost layer of stromal cells and collagen fibers, whereas the theca interna is going to be a highly vascularized internal layer, so a lot of blood vessels there, and we're also going to see this layer of cuboidal secretory cells. So you can see these cells here. We don't really need to name them here, but these cells are going to secrete estrogens. Now, also, in addition, those granulosa cells on the inside of the basement membrane, remember, part of that follicle, those granulosa cells are going to begin to secrete follicular fluid. Now, this follicular fluid is going to build up in a cavity that's called the antrum. So this is going to be in the center of a secondary follicle. So we can see that little cavity of fluid start to develop here, that antrum. So just further in words here, we have that secondary follicle. The theca is going to differentiate into the theca interna and the theca externa. And we also have this innermost layer of granulosa cells that's going to become really firmly attached to the zona pellucida. And that innermost layer of granulosa cells that's attached to the zona pellucida is now going to be called the corona radiata. Now that secondary follicle is going to mature and eventually become larger, turning into a mature follicle, also called a Graafian follicle. Now, while in this follicle and just before ovulation, that diploid primary oocyte is going to complete meiosis I. So it's going to complete meiosis I, and it's going to produce two haploid cells of unequal size. So we're going to have two haploid cells. These are just going to be labeled N and N, but they're going to be of unequal size. So one's going to be a lot bigger than the other. And each of these, because they are haploid, they're going to have 23 chromosomes. And like I said, that's going to happen within this Graafian follicle. Otherwise, everything is very similar to to what we saw with the secondary follicle. We have that antrum filled with follicular fluid. You can see the antrum is a lot larger than we saw in the secondary follicle. So those granulosa cells are going to be just producing more follicular fluid, creating that cavity to be a lot larger. And then we still have the same two layers of the theca folliculi. We have that zona pellucida and the corona radiata. Or to remember that corona radiata is going to be that layer of granulosa cells which is attached to the zona pellucida. Now remember I said that primary oocyte is going to go through meiosis I while it's in the mature follicle, and it's going to produce those two haploid cells. And like I said, they're going to be different sizes. So there's going to be a smaller cell that's produced by meiosis I, and we call this smaller one the first polar body. And essentially it's just going to be a packet of discarded nuclear material. It's not really useful. It's kind of just a garbage cell. Now the larger cell is going to be known as the secondary oocyte. So we're at a primary oocyte, we go through meiosis, and that produces a secondary oocyte. And this secondary oocyte is going to receive most of the cytoplasm. That's why it's going to be quite a lot larger than the first polar body. Now, once a secondary oocyte is formed, it does begin meiosis II, but it kind of stops in metaphase. It kind of pauses. Now, the mature follicle is going to soon rupture, and it releases its secondary oocyte, and this is a process that we know as ovulation. So here we have the process of oogenesis. So the top here, we're starting with a oogonium, and this cell is going to go through mitosis to produce a primary oocyte. And this primary oocyte goes through meiosis I to produce a secondary oocyte and that first polar body. Remember, this is a much smaller cell, just contains a lot of that excess material that the secondary oocyte doesn't need. It's going to be that secondary oocyte that is ovulated. It comes down here, and it's also going to be the one that is fertilized. So the secondary oocyte will become fertilized when it comes into contact with a sperm cell. And after it's fertilized, remember I said that secondary oocyte is going to start meiosis II, but then it kind of pauses it in metaphase. And it's only after fertilization that meiosis II is going to resume. So just kind of a good overview of the development of that oogonium. Here we have a really nice table that summarizes the events of oogenesis and follicular development. So here we have the age on the left column, oogenesis in the middle, and the follicular development on the right. So here it's also really cool how we can see how oogenesis and follicular development occur side by side, and we can also see where we have those connections. So again, we have the oogonium at the top turning into a primary oocyte, and that primary oocyte is going to develop with a primordial follicle. And then we get here after puberty, where that primordial follicle turns into a primary follicle and then develops into a secondary follicle. And then once that primary oocyte has developed into a secondary oocyte, that's when the follicle is a mature follicle or a Graafian follicle. So just a really nice table where you can kind of review everything that we've talked about so far with respect to oogenesis and follicular development. Now we're going to move on and talk a little bit more about the anatomy of the female reproductive system. So females have two uterine tubes. We also call these fallopian tubes or oviducts. Now these tubes provide a route for sperm to reach an ovum, and it also transports secondary oocytes and fertilized ova from the ovaries to the uterus. Now there's a funnel-shaped portion of each tube, which is called the infundibulum. Now this is close to an ovary, but it is open to the pelvic cavity, so it's not completely closed around each ovary. It essentially ends in these kind of like finger-like projections that we call fimbriae. And one of these fimbriae is attached to the lateral end of the ovary. The ampulla, this is going to be the widest and longest portion of the uterine tube. So we can see these anatomical structures here in this picture. So we can see these uterine tubes, one on either side of the uterus. So these uterine tubes will measure each about 4 inches long, and they lie within the folds of the broad ligaments of the uterus. So here we can see the infundibulum on either side. And from the infundibulum, the uterine tube is going to extend medially and inferiorly, and is going to attach to the superior lateral angle of the uterus. So again, this could be about right here and here. The ampulla, this makes up about the lateral 2/3 of the length of the uterine tube. And the isthmus, this is the more medial, very short, narrow, and thick-walled portion that joins the uterine tube to the uterus. Histologically, the uterine tubes are composed of 3 layers. We've seen these layers before: the mucosa, muscularis, and serosa. So that mucosal layer is going to consist of epithelium and lamina propria. Remember that lamina propria is going to be that areolar connective tissue. The epithelium within the mucosal layer is going to contain that simple ciliated columnar cells. And these are going to kind of function as a ciliary conveyor belt. And so it kind of really helps to move a fertilized ovum or a secondary oocyte within the uterine tube toward the uterus. So these simple columnar epithelium are really going to work as that kind of a ciliated, like I said, a ciliary conveyor belt. There's also going to be non-ciliated cells, which are called PEG cells, within the uterine tube. And these have microvilli. So they don't really act as that conveyor belt like the epithelial cells do, but these PEG cells do secrete a fluid that provides nutrition for the ovum. So here you can see the cilia of those ciliated simple columnar epithelial cells. You can also see these here. These are these long yellow cilia here. And again, remember, these cilia help to move the egg. So they kind of work as this conveyor belt to move the egg from the ovary all the way to the uterus. And then we can also see these PEG cells. Remember, these are non-ciliated, but they do have microvilli. On the right picture here, they are blue. And these help to nourish the ovum. The uterus, also sometimes called the womb, serves as part of the pathway for sperm that's deposited in the vagina to reach the uterine tubes. This is also going to be the site of implantation of a fertilized ovum. This is also where we have development of the fetus during pregnancy and labor. Now, during the reproductive cycles, when implantation of a fertilized ovum doesn't occur, the uterus is going to be the source of menstrual flow. Anatomical subdivisions of the uterus are going to include the fundus, which is going to be the dome-shaped portion, which is superior to the uterine tubes. We also have the body, which is kind of this tapering central portion. And the cervix. So this is the inferior narrow portion that opens up to the vagina. And then between the body of the uterus and the cervix, we have the isthmus. And this is a constricted region. It's about 1 centimeter long. The interior of the body of the uterus is called the uterine cavity. And the interior of the cervix is called the cervical canal. The cervical canal opens into the uterine cavity at the internal os, and then it opens up into the vagina at the external os. Now, histologically, the uterus consists of 3 layers of tissue. First is the perimetrium, then we have the myometrium, and the endometrium. So these are going to be your 3 layers of tissue within the uterus. The outermost layer, the perimetrium, this is part of the visceral peritoneum. Then the middle layer of the uterus, the myometrium, consists of 3 layers of smooth muscle. So this is going to be the muscular layer. And then the inner layer of the uterus, the endometrium. This is very highly vascularized and it's divided into 2 layers. So the 2 layers of the endometrium are the stratum functionalis and the stratum bassalis. So the stratum functionalis, also known as the functional layer, this lines the uterine cavity and pretty much sloughs off during menstruation every month. The deepest layer, or the stratum bassalis, also known as the basal layer, this is a permanent layer and it gives rise to new stratum functionalis after each menstruation. So this layer doesn't slough off, or it doesn't peel off, it doesn't slough off every month. This is a permanent layer, whereas the stratum functionalis sloughs off every month, or rather every menstruation. Now you can't see it on this section, but the perimetrium is composed of simple squamous epithelium and areolar connective tissue. You can see the myometrium here though, and this again consists of 3 layers of smooth muscle fibers that are going to be thickest in the fundus and thinnest in the cervix. Just kind of an interesting fact, not something that you actually have to remember. The inner layer of the uterus, the endometrium. This is again going to be highly vascularized and consisting of those 2 layers, the stratum functionalis that sloughs off during menstruation, and the stratum bassalis. The secretory cells of the mucosa of the cervix produce a secretion which is called cervical mucus. And this is a mixture of water and glycoproteins, lipids, enzymes, and even some inorganic salts. Cervical mucus is more hospitable to sperm at or near the time of ovulation because it's at that point that the cervical mucus is less viscous and it's more alkaline. So it actually holds a pH of about 8.5 during ovulation. Now this mucus will supplement the energy needs of the sperm, and both the cervix and the cervical mucus will protect the sperm from phagocytes and also protect them from the really hostile environment of the vagina and the uterus. Cervical mucus may also play a role in capacitation. This is a really important term that we definitely need to know. So capacitation is a series of functional changes that sperm will undergo in the female reproductive tract before they're able to fertilize a secondary oocyte. So really what capacitation does is it causes a sperm cell's tail to beat even more vigorously, and it prepares the sperm cell's plasma membrane to fuse with the oocyte's plasma membrane. Capacitation essentially allows the sperm to fertilize a secondary oocyte. It gives it all the tools it needs. Okay, moving on to the vagina. So the vagina is a tubular, about 4-inch long fibromuscular canal. Again, it's lined with mucous membrane that extends from the exterior of the body to the uterine cervix. So we could see it here, going from the cervix down here to the external orifice. The vagina is the receptacle for the penis during sexual intercourse. It's also the outlet for menstrual flow and also the passageway for childbirth. Now, situated between the urinary bladder and the rectum, the vagina is directed superiorly and posteriorly where it attaches to the uterus. So here we can see, again, we can see the rectum here. We can see the uterus kind of goes up above the bladder. So we have the uterus up here, and then the bladder here. The mucosal layer of the vagina is continuous with that of the uterus. Histologically, it consists of these non-keratinized stratified squamous epithelium and also areolar connective tissue we know as lamina propria. So we have these two main layers of tissue in the vagina. We have the mucosal layer. Again, remember this is made up of these non-keratinized stratified squamous epithelium and lamina propria. And we have a muscularis layer. There's also the adventitia, but again, the main two layers are just going to be mucosal and the muscularis layer. Okay, now the epithelium and the areolar tissue within that mucosal layer are going to lie in a series of transverse folds that we call rugae. Again, these are going to be kind of similar to the rugae that we see in the stomach. Then we have the muscularis layer, and this is composed of an outer circular layer and an inner longitudinal layer of smooth muscle. And these layers of smooth muscle can stretch considerably. This is to accommodate the penis during sexual intercourse and also to accommodate a child during birth. A thin fold of vascularized mucous membrane, which is called the hymen, this forms a border around and partially closes the inferior end of the vaginal opening to the exterior, the vaginal orifice. The term vulva, also called pudendum, this refers to the external genitals of the female. So these following components are going to make up the vulva. First, we have the mons pubis. This is just anterior to the vaginal and urethral openings. And this is an elevation of adipose tissue that's covered by skin and coarse pubic hair that cushions the pubic symphysis. Now, from the mons pubis, there are two longitudinal folds of skin, which are called the labia majora, and these extend inferiorly and posteriorly. Now, just medial to the labia majora are two smaller folds of skin called the labia minora. Next, we have the clitoris. This is a small cylindrical mass composed of two small erectile bodies. And the region between the labia minora is the vestibule. Now, within the vestibule, we have the hymen, the vaginal orifice, the external urethral orifice, and the openings of ducts of several different glands. Okay, so here are some of those glands that we're going to go over. On either side of the external urethral orifice are going to be the openings of the ducts to the paraurethral glands. These are also called Skene's glands. Now, these are mucus-secreting glands, and they are embedded in the wall of the urethra. The paraurethral glands are homologous to the prostate. Now, on either side of the vaginal orifice itself, we have the greater vestibular glands, also called Bartholin's glands. And these open by ducts into a groove between the hymen and labia minora. Now, they produce a small amount of mucus during sexual arousal and intercourse, and they are going to be homologous to the bulbourethral glands in males. Then we have the bulb of the vestibule. Now, this consists of two elongated masses of erectile tissue that are just deep to the labia on either side of the vaginal orifice. The bulb of the vestibule becomes engorged with blood during sexual arousal, which narrows the vaginal orifice. The bulb of the vestibule is homologous to the corpus spongiosum and the bulb of the penis in males. So I'll just put this here. I do believe that there's a table a little bit later that shows all the homologous structures between males and females yet. This table here summarizes the homologous structures of the female and male reproductive systems. So here we can see things like the ovaries in the females are homologous to testes in the males. Also, some important ones are going to be the vestibule in females is homologous to the intermediate urethra in males, the bulb of the vestibule in females, homologous to the corpus spongiosum penis and the bulb of penis in males. And then also these glands here, the paraurethral glands, homologous to the prostate in males, and then the greater vestibular glands in females being homologous to the bulbourethral glands in males. Okay, we're going to talk about some other accessory structures in the female reproductive system. Now, within each breast, there is a mammary gland. Now, mammary gland is a modified sudoriferous gland or sweat gland, and these produce milk. Now, a mammary gland contains about 15 to 30 lobes or compartments, and these are going to be separated by kind of a variable amount of adipose tissue. In each lobe, there are several smaller compartments that we call lobules. And these lobules are composed of kind of like grape-like clusters of milk-secreting glands that we call alveoli. And these alveoli are going to be embedded in connective tissue. When milk is being produced, it passes from the alveoli into a series of secondary tubules and then into the mammary ducts. These ducts, we call them lactoriferous ducts. These will each emerge through a nipple. So here we can see that one pigmented projection called the nipple, and this has a series of closely spaced openings of ducts that we call these lactiferous ducts, and this is where the milk emerges from. The circular pigmented area of skin that surrounds the nipple, this is called the areola. And this appears kind of rough because it contains those modified sebaceous glands or oil glands. Now, strands of connective tissue called the suspensory ligaments of the breast, we also call these Cooper's ligaments. These run between the skin and the fascia, and these support the breast, kind of hold it in place. During their reproductive year, non-pregnant females will normally have these cyclical changes in the ovaries and the uterus. Each cycle takes approximately 1 month. Again, everyone is different, but approximately it's a month. And it involves both oogenesis and the preparation of the uterus to receive a fertilized ovum. Hormones that are secreted by the hypothalamus, the anterior pituitary, and the ovaries will control the main events of these cycles. The ovarian cycle, this is going to be the first one we're going to talk about. This is going to be a series of events in the ovaries that occur during and after the maturation of the oocyte. The uterine cycle, we also call this the menstrual cycle. This is a series of changes in the endometrium of the uterus, and these are all changes that will help to prepare the uterus for the arrival of a fertilized ovum, and this ovum will develop there until birth. Develop into an embryo and then a fetus and then a baby. Now, if fertilization doesn't occur, these ovarian hormones will wane, they'll decrease, and this causes the stratum functionalis of the endometrium to slough off. Now, the general term female reproductive cycle, as we can see in the title here, this kind of encompasses both the ovarian and uterine cycles. It also includes the hormonal changes that regulate these cycles, and the female reproductive cycle also includes the related cyclical changes in the breasts and cervix. So this whole female reproductive cycle term kind of encompasses kind of a lot of things. The gonadotropin releasing hormone, GnRH, this is secreted by the hypothalamus. This is going to be the main hormone that controls the ovarian and uterine cycles. The gonadotropin releasing hormone stimulates the release of the follicle stimulating hormone and also the luteinizing hormone. These are both from the anterior pituitary. So that GnRH stimulates the release of these two main hormones. So this figure here shows the secretion and also the physiological effects of estrogen, progesterone, relaxin, and inhibin. And this is going to be in the female reproductive cycle. So remember, we kind of saw this figure a little bit before with the gonadotropin releasing hormone being released from the hypothalamus, causing the secretion of the follicle stimulating hormone and luteinizing hormone in the anterior pituitary gland, stimulating the follicular growth here, ovulation, and then also the corpus luteum being created. So we're going to talk about just a little bit about these 4 hormones at the bottom and kind of what their functions are. So first we have estrogens on the left here. These estrogens are going to be secreted by ovarian follicles, And they do have several important functions. So first off, they promote the development and maintenance of the female reproductive structures. They also help with the development of feminine secondary sex characteristics and also the development of the breasts. Estrogens will also increase protein anabolism. So this includes the building of strong bones. So protein anabolism. Estrogens are also going to help to lower blood cholesterol levels. And they also help to moderate levels in the blood to inhibit both the release of GnRH by the hypothalamus and the secretion of LH and FSH by the anterior pituitary. So those are 3 hormones there. Next we have progesterone. This is mainly secreted by the cells of the corpus luteum, and progesterone really kind of cooperates with estrogens to prepare and maintain the endometrium for implantation of a fertilized ovum, and also helps to prepare the mammary glands for milk production and secretion. The small quantity of relaxin that's produced by the corpus luteum during each monthly cycle helps to relax the uterus by inhibiting the contractions of the myometrium. That's that uterine smooth muscle. So that's going to be the main thing that relaxin does. During labor, it has a lot of other functions, but we're mainly just going to talk about the role of relaxin just in the monthly cycle when there isn't fertilization and implantation. Then we have inhibin. This is secreted by the granulosa cells of the growing follicles and also by the corpus luteum after ovulation. And the main function of inhibin is going to inhibit the secretion of the follicle-stimulating hormone and, to a lesser extent, the luteinizing hormone. Now, the duration of the female reproductive cycle typically ranges about a month, so like I said, but here about 24 to 36 days. Now, just for simplicity's sake, we're just going to assume a duration of about 28 days, and we are going to divide it into 4 phases. So these 4 phases are going to be the menstrual phase, the pre-ovulatory phase, ovulation, and the post-ovulatory phase. So here we have the female reproductive cycle in a diagram. We have the ovarian cycle here in the middle and the uterine cycle at the bottom. We can see the endometrium here, the stratum functionalis layer and the stratum basalis layer. And then at the bottom, we can see how many days are in the cycle. So starting here at 1 and going till day 28. So we're going to kind of talk about this, how they all connect to one another and the events that occur during the cycle. So first we have the menstrual phase down here in the bottom left. This is also called menstruation. And this lasts for roughly about the first 5 days of the cycle. By convention, we're going to say that the first day of menstruation is going to be day 1 of a new cycle. Now, under the influence of the follicle-stimulating hormone from the anterior pituitary gland, several of these primordial follicles are going to develop into primary follicles and then into secondary follicles. Again, this is just a bit of a review when we talked about the ovarian cycle earlier. Now, at the same time, menstruation or menstrual flow from the uterus is going to kind of consist of about 50 to 150 milliliters of blood and tissue fluid, mucus, and epithelial cells from that are all shed from the endometrium. Now, this discharge occurs because of the declining levels of progesterone and estrogens, and it stimulates the release of prostaglandins that causes these little spiral arterioles in the stratum functionalis here, causes them to constrict. So prostaglandins will cause these uterine spiral arterioles to constrict, and as a result, the cells that these blood vessels supply will actually become oxygen deprived and start to die. So that's kind of what menstruation is caused by. Then next we have the pre-ovulatory phase. Now this is really just the phase between the end of menstruation and up until ovulation. Now this phase of the cycle is more variable in length than the other phases, and this is going to be the phase that really accounts for most of the differences in length of cycle between different people. Now some of the secondary follicles in the ovaries begin to secrete estrogens and also inhibin. Now by about day 6 here, we have a single secondary follicle in one of the two ovaries has outgrown all of the others, and it has kind of become the dominant follicle. Now normally there's going to be one dominant follicle that becomes the mature follicle. Now this mature follicle continues to enlarge until it's more than 20 millimeters in diameter, and it's going to be ready for ovulation. Estrogens that are liberated by that growing ovarian follicles will stimulate the repair of the endometrium. So we kind of get this proliferative phase. So the endometrium is going to thicken, it's going to repair, and as a result, the endometrium in this phase doubles in size, or approximately doubles in size. Then we have ovulation. So this is where we have that rupture of the mature follicle and the release of the secondary oocyte into the pelvic cavity. And usually ovulation occurs on day 14 in a 28-day cycle. Lastly is the post-ovulatory phase. This is the time between ovulation and the onset of the next cycle. Now, after ovulation, the mature follicle collapses on itself, and the basement membrane between the granulosa cells and the theca interna is going to break down. The theca interna cells will mix with the granulosa cells as they become transformed into the corpus luteum. And this is usually created, this corpus luteum is really created under the influence of the luteinizing hormone, again from the anterior pituitary. Now, as the corpus luteum is continued to be stimulated by the luteinizing hormone, the corpus luteum secretes progesterone, estrogens. It also releases some relaxin and inhibin. So the corpus luteum secretes all 4 of these hormones that we talked about just previously. Now the progesterones and estrogens that are produced by the corpus luteum will promote growth and coiling of the endometrial glands. It'll also promote vascularization of the superficial endometrium. So we're going to get those blood vessels all the way to the surface here. We have, I'll use different color here. We have these glands here. And we're also going to get thickening of the endometrium in this phase as well. So that's going to be caused by the progesterones and estrogens that are produced by the corpus luteum. So I'm going to go these are going to cause 3 things. So first is going to be promoting that growth and coiling of the endometrial glands. I just put endoglands, but these are the endometrial glands. We're also going to get vascularization of the superficial endometrium. So I'm just going to put vascularization. So just extending those blood vessels more superficially. And the third thing is going to be thickening of the endometrium even further, up to about like 12 to 18 millimeters. So this is going to be the thickest at this phase. So we're just going to put endometrial thickening. Now, this graph here gives us a really good look at those changes in the anterior pituitary and ovarian hormones during that whole female reproductive cycle. So in the menstrual phase, remember that's about day 1 to 5 in the cycle, we have the follicle stimulating hormone, the blue line here. This is going to influence several of those primordial follicles to develop into primary follicles and then into secondary follicles. Remember, it's here that we have pretty low levels of estrogens and progesterone. As we get to around day 10 to about day 12, we're kind of during that final maturation process in the pre-ovulatory phase. This is where the mature follicle continues to increase its production of estrogens. So we can see this green dotted line that represents estrogen levels really start to increase in this final stage of the pre-ovulatory phase. Now, the high levels of estrogens during the last part of that pre-ovulatory phase are going to exert a positive feedback effect on the cells that secrete the luteinizing hormone and cause ovulation. So we're going to get that really high levels of luteinizing hormone right before ovulation that causes ovulation right around day 14. And then we have that post-ovulatory phase after ovulation where the corpus luteum secretes a lot of progesterone and estrogens. You can see this green line here, those estrogens. Remember that the corpus luteum is also going to secrete relaxin and inhibin, which aren't on this diagram, but the corpus luteum does secrete all 4 of those. Now, later events in an ovary that has ovulated an oocyte really depend on whether the oocyte is fertilized. So if the oocyte is not fertilized, the corpus luteum will last about 2 weeks. Then its secretory activity declines and it degenerates into pretty well like a scar tissue that we call corpus albicans. Now, as the levels of the progesterone and estrogens and inhibin will decrease at the end of the cycle here, the release of gonadotropin releasing hormone, follicle stimulating hormone, and the luteinizing hormone will rise. We can see that here, they're starting to go up already here, the follicle stimulating hormone and luteinizing hormone. And this is due to a loss of negative feedback suppression by the ovarian hormones. So the progesterone and estrogen were kind of having a negative feedback on FSH and LH, but as they decline at the end of the cycle, that negative feedback suppression kind of goes away and the LH and FSH will start to rise again. And then this is going to signal kind of the beginning of a new cycle. Again, this is only if fertilization has not occurred. We're going to start a new cycle again. As we've kind of learned from this entire class, feedback is important really everywhere in the body. So like I said before, high levels of estrogens during that last part of the pre-ovulatory phase are going to exert a positive feedback effect on the cells that secrete the luteinizing hormone and gonadotropin releasing hormone, and this causes ovulation. So here we have a diagram of the events leading up to ovulation. So kind of numbered here, we'll start at number 1, where a high concentration of estrogens will stimulate more frequent release of the gonadotropin releasing hormones from the hypothalamus. Now, it also directly stimulates the gonadotropes in the anterior pituitary to secrete more luteinizing hormone. Now, the gonadotropin-releasing hormone, number 2 here, this is going to promote the release of follicle-stimulating hormone and luteinizing hormone from the anterior pituitary. And that luteinizing hormone is really just going to stimulate or cause the rupture of the mature follicle here at the bottom. So we have this mature follicle here at the bottom. And that luteinizing hormone in number 3 is going to cause the rupture of that mature follicle and the expulsion of the secondary oocyte about 9 hours after the peak of that luteinizing hormone surge. Then the ovulated oocyte and its corona radiata cells are usually going to be swept into the uterine tube. So this is just the events of ovulation here. So this diagram may look pretty big and confusing, but really, I promise we've covered all of this already. So this is a diagram summary of the hormonal interactions in the ovarian and uterine cycles. This is a really helpful figure, really just shows everything that we've talked about so far, kind of just in one diagram here. So we have the hormones coming from the hypothalamus and anterior pituitary at the top here. We have the ovarian cycle in the middle, and then the uterine cycle at the bottom here. Positive feedback is shown in green arrows, and negative feedback is shown in the red dashed arrows. So just like every other system in the body that we've talked about, we definitely want to look at how the reproductive system interacts with other systems in the body. So just a couple things to note here. In the skeletal system, androgens and estrogens will help with bone growth and also maintenance of those bones. In the muscular system, those androgens will also stimulate the growth of skeletal muscles. In the nervous system, estrogens can also play a role in development of certain brain regions in males. And we already briefly talked about this or briefly mentioned this, but in the cardiovascular system, those estrogens will help to lower blood cholesterol levels, can also help to reduce the risk of coronary artery disease. So all important things here. And that's the end of chapter 27.